My name is Jaime Huerta Cepas. I'm working in the Comparative Genomics lab in Madrid, in the CBGP Institute, and I'm the lead developer of the ETE Toolkit. ETE Toolkit, the letters ETE stands for Environment for Tree Exploration. So basically, we are maintaining software to deal with trees in general, although our main focus is on phylogenetic trees because this is our main research topic, and especially on visualization. And the funds that we got from this initiative were specifically intended to push a new version of the ETE Toolkit, including new features. And one of these features, probably the main one, is improving the visualization of very large phylogenetic trees or trees in general. So let me first introduce a little bit what is ETE Toolkit and what kind of things you can do with it. So essentially, ETE Toolkit provides a Python library that you can import in your own scripts and interact with and use it to interact with trees. So you can load trees in NeWik format, for instance, and then you can start operating with the tree as a normal Python object. And you can traverse, search, you can modify the topology, you can compare trees, you can do a lot of stuff programmatically. Then ETE also provides a way of annotating different nodes in the trees. Annotating means that you can add any kind of feature that you want to, or characteristic to the different nodes, and then you can use all those features to analyze the tree or even visualize. This is one of the key components of the ETE Toolkit also, that you can operate with the trees in a visual manner, also in a programmatic way, meaning that the same scripting language that we are using in ETE to operate with the tree, you can use it to program how you want to visualize this tree, taking care of all the annotations that you want to visualize and integrate in the visualization. It also provides some Jupyter integration, so it's very dynamic and interactive. So this is the essential part of the ETE Toolkit software. Apart from this, there are other tools, so to say, so that they are not providing an API. They are just scripts and tools that you can use from the command line to run specific analysis. In particular, as I said, we are very focused on phylogenetics, so there are many tools to run phylogenomic analysis, like automated reconstruction of gene and species trees. You can run some evolutionary test hypotheses. ETE also provides a way of connecting your trees with taxonomic databases like the NCBI database. You can compare trees, you can summarize information in different trees, etc. But the part that we are currently working on very actively is in how to connect all those features and annotations and analysis that you do with the trees and the programmatic visualization language that we have in ETE. So we want to improve the visualization so it allows us more options, and especially to scale with many other, with the kind of data sets that we are dealing with in the last years. So ETE is very flexible. It allows you to programmatically control every aspect of the tree image that you are generating. All these images that you see here are generated with small scripts programmed in Python and using this language that I was mentioning. And to do so, ETE uses, I don't know how to say, there is a philosophy on how this visualization is somehow organized in the ETE language. So there are three basic layers that you can use to control programmatically how the trees are being visualized. So one is that you can attach to any tree object a tree style object, and with this you can control very easily how you want to plot the tree. If you want a circular representation, rectangular, if you want rotating, if you want to reduce the angle, and many other aspects. Then there is another layer that you can also dynamically control, which is how you want every node in the tree look like. So you can access different properties of the nodes in every tree and set from size, color, any visual attribute that you want to control, you can do it per node. And because you are doing this programmatically, you can attach this configuration, this visual configuration to conditions in your script. So this makes very dynamic the process of automating the visualization. And finally, and this is a very, in my opinion, very powerful feature of the programmatic visualization system in ETE, you can attach decorations to every node. So you can not only control how, the color and the size of the node, you can also attach custom elements, decorations to every node. And for this, ETE uses an abstraction, which is that every node has a virtual grid where you can put elements. And this virtual grid is controlled by a very basic vocabulary. So you can create a small graphical item and you decide where you want to put it relative to the position of the node. So if you are decorating one node and you want a text label over the branch of the node, then you can assign this to a specifically top branch position in the column and the row that you want. And using this abstraction and the helper functions that are available in ETE, you can control dynamically what elements, what decorations are being attached to every node and how they should be plotted. Everything programmatically, as I said. And this is very powerful. You can create all those example images that I showed you before with this system. So this is just a very simple, this is actually a full example where with a few lines of code, you can create something, an image like the one that you see on the right here. So you control every aspect programmatically. So what was the problem? This model has been working very well for us, working on tree analysis and visualization in the lab for analytical purposes. So we are visualizing trees all the time on a daily basis, and this system is working quite well in general. The problem is that the data sets that we are starting to deal with in the genomics area are huge. The bacterial phylogenies that are being generated grow exponentially, and just the tree of life that you would like to represent at some point is orders of magnitude larger than the size of trees that ETE can handle from a graphical perspective. So this is a big problem for us, and it has been a problem for a few years already. So basically, what I'm showing here in this slide is that what we want to port to the lab is something what you see here in this image. So basically, we want something really flexible and easy to use that we can use to visualize any tree, any large tree that we are generating in the lab. And by large, I mean really huge trees. So if usually you visualize trees of a few thousand tips, we are talking about trees of million nodes that we would like to visualize at some point. There are some notable solutions so far being developed, like the Tree of Life browsers that were published in the last years. So one is OneZoom, the other is LifeMap. These are very nice solutions, very artistic also, and it allows you to browse the Tree of Life in a graphical way, but still it's not the type of analytic plotting that we are looking for. So we want to connect these technical advances with the programmatic system that we provide with the ETE software. So there are two main problems. One is that the programmatic tree visualization is uncommon in software. Fortunately, we have this covered in the ETE Toolkit. And the second problem is that the viewers, I mean, the interactive viewers are not optimized for large tree exploration. So it is hard to go from above 5,000 nodes in the tree and have smooth interactive browsing of trees. The GUIs become unresponsive, the layouts are not readable, and a lot of features associated with phylogenetic trees are usually not interpretable, like branch lengths, etc. So we set basically a challenge a few years ago, and I basically started playing with the code and see if we can port ETE to something like this. So I set this challenge. I wanted to get smooth, responsive, and interactive exploration of a programmatically annotated phylogeny. This means that I want many decorations in this tree, but of a tree with 1 million nodes, at least 1 million nodes, using only 1 GB of RAM, 1 CPU core, and at least moving this tree interactively at 1 frame per second, which is very low, but still fine for our purposes. So I ended up, I tested a lot of approaches, and I ended up having a really hard fork of the ETE code. Now, I mean, this is where we are, and we have some prototypes. The purpose of this project here in this funded by this initiative is to port all these prototypes into something workable and useful in the next version of ETE. So I'm going to briefly mention what are the challenges that we somehow overcome with the prototypes and that we are porting at the moment in the ETE code. So the first strategy that we use is that we are not drawing the whole tree at once. We are being smart and we are collapsing and expanding nodes depending on the zoom level. And this is quite easy to get, and it provides a cleaner scene and even more responsive graphical user interface, etc. So this is a short video showing how our current prototype works, and you can see this is 1,000 leaves, so it's not super large, but you can see that you can get very responsive graphical user interface. The second challenge that we wanted to overcome was how to have a clean layout. So when you are exploring large trees, it's very likely that you are overlapping decorations, and suddenly the visualization is not practical because you have everything on top of everything. So now we have a prototype that is providing you adaptive drawing of those node decoration, meaning that when you are zooming in and zooming out, the decorations are also collapsed, hidden, or shown depending on your zoom level. So if you see, not only the decorations here are being hidden or shown, they are also adapted in size and in format. So this is also part of the working prototype. There is another big problem that we had exploring large trees, derived from the fact that branch lengths are important for us. And when you are visualizing a very large tree where the branch lengths are important, then you have this kind of problems where at the very zoom, when you are looking the tree from the outside, then the branch lengths are maybe readable, the basal branch length. But when you zoom in, even if you can zoom in very smoothly, the branch lengths are not interpretable. And you can see this with the next comparison. So this is the same tree on the left. You are seeing how it looks when you are zooming in a tree, a large tree, without correcting the branch lengths. And on the right, you see what it happens if we automatically adapt the branch lengths depending on the zoom level. And this is also part of the prototype that we are working on at the moment. So there you can browse very large trees, and the scale of the things that you have on screen is comparable, but is adapted to the zoom level where you are. The same strategy for branch lengths and adaptive decorations. We are also using it to have something that we call information-aware zooming, meaning that we are not only showing or hiding stuff when you are zooming in, zooming out. We are also interested in summarizing the information. So if you are browsing a tree of 1 million tip nodes, then you want to have some information of the node that is grouping 500,000 of those nodes. So this is also something that we can control dynamically. So when you are zooming out or zooming in, the annotations and the decorations that you see represent the summary of the information that is underneath those nodes. So this is another example of how it looks. So here the annotations are this heat map that you see in the outer circle. So when you are zooming in, you will see the outer circle, the heat map is being adapted. So you can see what are the values associated to to individual items. But when you are zooming out, what you see is the summary of all the items that are grouped by the different nodes. And this is already a tree of 100,000 tip nodes. This is already huge. Not many software for visualizing trees can even move these trees. And with the prototype that we are working on at the moment, we can not only move the tree, we can also adapt dynamically all these annotations. So how large can we go with this? So we have been doing some tests. Recently, this is also based on the prototype that I was mentioning. This is 10 million nodes. So this is really huge. We don't even have a data set like this yet. And still we can move it with only 7 gigs of RAM, 1 CPU, and just 5 minutes of preloading all the information that we need. And then the browsing, it doesn't matter how big the tree is. The browsing is always smooth, and you can browse in, browse out, zoom in, zoom out very, very smoothly. So this is where we were when we applied to this grant with this initiative, and we got funds to convert this into something real and implemented it into the ETE Toolkit code. So this is my last slide. So this is just summarizing what is the purpose and what we are trying to do at the moment with all this code. So basically, we are refactoring all those prototypes into something that is compatible with the base code of the ETE Toolkit. And we are also working on a web implementation. So all you have seen so far is a standalone version based on Qt libraries, but we want to have some web-based implementation so other projects can also use it and improve, of course, the type of decorations, etc. This is something that now we are advancing really fast, I must say, with these funds that we received, because Jordi joined the team and he's really working very fast and trying to get something useful as soon as possible, and we hope to have it in the following month. So if you are interested, just follow the news on any of these accounts, and we will be happy to share what we are doing. Thanks a lot.
Here we go. Sean Goggins has a question. So he asks, when you say hard fork, I assume the intention is that it will remain in that fork. Is the work you showed on GitHub as of yet?
Yes, I mean, the reason it's not in GitHub yet is because it's a bit embarrassing, the code that we have there with all the prototypes and tests, etc. So Jordi is specifically taking care of this and is extracting the good code or the code that is actually working, porting to a new branch, and we will upload to GitHub as soon as it is decent, at least, to share.
We have all been there. I think we have time for just one more question. Any remaining curiosities? All right, so with that, let's thank QIIME once more. I'm loving these little reactions. And then we'll move on to our second speaker, and I believe, so GATK, yes. So the floor is yours.
Hello, everyone. My name is Bhanu Gandham. I am a bioinformatician at the Broad Institute. I lead a team that is responsible for the adoption and optimal use of GATK tools. Today, I'm going to be presenting on our project called GATK for Bacteria. The focus of this project is to develop methods for variant discovery in diverse bacterial species. So in today's talk, I will cover 3 main parts. One, I will give you a brief overview of the background and the motivation of why we got this project started. I'll talk about the 2 variant discovery methods that we've worked on. One is single reference based and the other is pan-genome based. And then I will touch a little bit on our community adoption steps and some of the and a slight glimpse into some of the future work that we've planned for this project. GATK stands for Genome Analysis Toolkit. This toolkit has more than 240 tools and over 75,000 users worldwide. However, this toolkit was primarily designed for human variant discovery purposes. In my current role at the Broad, I interact with researchers very closely, and I've been asked multiple times about how to use GATK for microbial analysis. And my answer has always been, well, we haven't tested it out on our end, but why don't you try it out and let us know and provide us feedback? And I said that so many times that I got tired of saying it. And so I decided to do a little bit of research of my own, because GATK was never intended for microbial analysis. And what I realized was not only are researchers using GATK for microbial analysis and getting results, they're also publishing papers on it. And one such example is SnapperDB. SnapperDB is a database of variant data from bacterial isolates. And this database used GATK to discover these variants. And not only that, there are all of these other analyses that are using SnapperDB for their purposes. For example, the surveillance of listeriosis, this other analysis, which is about characterization of E. coli, characterization of rare salmonella, public health risk assessment reports, and outbreak studies. And there are 23 more such papers which talk about using SnapperDB, which indirectly uses GATK for microbial analysis. And all of this was happening without us intending for it to happen, without GATK intending for it to happen. And so we thought the responsible thing to do here would be to do some benchmarking, figure out what GATK is doing right, what it's doing wrong, and fix it. And that's where CCI came in and provided us this grant to pursue this project. So why bacteria? Why did we start with optimizing GATK for bacteria? Because antibiotic-resistant bacteria is a major global threat. In US alone, there have been more than 2.8 million antibiotic-resistant infections and more than 35,000 deaths. And this is just in US alone. In developing countries, the situation is much worse. In a 2019 CDC report, Robert Redfield, who's the director of CDC, said, "Stop referring to a coming post-antibiotic era. It's already here. The time for action is now, and we can be a part of the solution." And so it is critical that we provide the research community with robust tools to study how bacteria develops resistance and how it spreads. And so with that idea in mind, we came up with this three-pronged approach for GATK for bacteria. One, we wanted to develop open source, scalable, portable, interoperable, and reproducible software. Two, we want researchers to be able to use this software to study how bacteria evolves, how it develops resistance and causes diseases. And finally, we want to package this software in such a way that researchers are able to share their data and analysis and be able to collaborate on a global scale. And so why is it important for us to optimize GATK for bacteria? The reason for that is, as I mentioned earlier, human genome is drastically different from bacterial genomes. Human DNA is linear and diploid, while bacterial is circular and haploid. Human DNA has true variants in varying read depth regions of the sequence genome, and artifacts that are specific to bacteria, but you don't see this in human DNA or human genomics data. And also, bacterial genomes are highly diverse. For example, if you take any two samples of human samples, they're about 99.9% similar. However, for example, E. coli bacteria, if you take any two strains, they are no more similar than 50% of their genome. So before I go into some of the implementations for how we optimize GATK for bacteria, let me give you a little overview of how GATK works under the hood, how the variant caller works. GATK assumes and recognizes that aligners can make mistakes. They're not always going to align reads with perfection. And so it does its own local assembly. And so how it does that is, first, it identifies regions in the genome that have potential variations, and these are identified as active regions. It then does local assembly in this region and creates a graph from which it determines what are some potential haplotypes. And then it maps all the reads to these haplotypes And based on that, it determines what the likelihood of each variant is and the likelihood of each genotype. And so what you're looking here now is an IGV screenshot of an example of the optimization that we implemented in GATK. So the track right on top is GATK out of the box without any optimizations for microbes. And you can see that the first track is showing no variation in this region. However, the second track shows that there's an indel event in this region. From Illumina reads, you can tell that there is some variation. Nanopore, we also compared Nanopore and PacBio data, and you can see that Nanopore and PacBio support the evidence of an indel in this region. Even though GATK for bacteria pipeline doesn't identify this large indel events that you're seeing in Nanopore and PacBio, which is because of the limitations of short reads data, it is still doing a good job of identifying that there is some kind of an indel event happening in this space. So why didn't GATK out of the box identify this? The reason for that is dangling ends. Because as you can see here, there are reads that above and below the reads with variation, there are reads with very low mapping quality, and there's not enough evidence to create a contiguous graph. And so it ends up with these dangling ends. This in human data, if you find such dangling ends in human data, these are mostly signs of artifacts. But because bacterial genomes are so diverse, this kind of occurrence is very common, and there are lots of indels you see in bacterial data, which end up with such dangling ends. And so it's important to optimize and recover these dangling ends, which is what we have done in our GATK for bacteria optimization. Some other improvements that we've implemented is we have two variant callers in GATK. We have a germline and a somatic variant caller. We've noticed that most researchers use the germline variant caller, but we have noticed that our somatic variant caller called MUTEC2 has much higher sensitivity for bacterial variant calls. And also because the germline variant caller determines variant likelihood depending on the ploidy and its associated allele frequencies. And so we've based on our benchmarking, we've noticed that our somatic variant caller does a much better job. In addition to that, as I mentioned earlier, bacterial genomes are circular in nature. And so for sequencing purposes, when they're linearized, there's an artificial breakpoint that's created. And any reads that span those breakpoints are lost. And so we've developed a tool that helps capture reads that span this breakpoint. In addition to that, we've also optimized for better filtering of bacterial variants, as I've shown you before. There are variants that you see in bacterial data, which in human data may be an artifact, but in bacterial data, it's very common. And so we had to tweak a lot of the filtering steps as well. So another main challenge with bacterial data, as I mentioned before, is that it has this high diversity, right? So what you're looking at here is when you map, say, the reads from a sample to the reference. Now, if your reference does not have all the regions that are actually present in your sample strain, then it's just going to assume that these are artifacts or these regions don't exist in this sample strain. It'll drop reads that map in this particular region. And so any variation in these regions will not be called if you're comparing it to a single reference. And this is, again, because of the high diversity in bacterial species. And so as a workaround, we've prototyped a tool which essentially creates a pan-genome graph from all the references that are available for a particular strain. So for example, E. coli has about 1,000 different strains in RefSeq. So we create a gigantic graph of all the references, and then we map the sample against this reference. So now the regions that you had previously seen on the previous screen, which you which didn't have where we were not able to identify any variance because it wasn't present in the closest reference, it is now being able to be captured because we have now introduced other references to the graph. And so, like I said, we're now able to identify all of these variants, which we wouldn't have been able to identify with a single reference. And so this is one example of what we were able to identify using our pan-genome approach. We were able to identify this 1.1 kb chunk that was present in our sample, but wasn't present in our closest reference. And if not for the pan-genome method, this 1.1 kb chunk would have gone unidentified. But this chunk was present in 200 other E. coli genomes. And also, this chunk is actually beta-lactamase. Beta-lactamase is important because beta-lactamase is a gene that, that is associated with beta-lactam antibiotic. And this gene prevents this bacteria, it actually helps the bacteria to become immune to the beta-lactam antibiotics. And so I've talked a lot about all the optimizations we've done in GATK to improve its usability in microbial species. But as long as we don't make these tools and software available to the community and make it available in such a way as to allow for ease of adoption, it's not going to make the impact that we want to make. We want researchers to be able to share their data analysis, and we want researchers to be able to collaborate on a global scale. And so for that purposes, we have written all of our tools in this workflow descriptive language called WDL, which makes these workflows platform agnostic. We've also provided these WDLs, these workflows in Terra, which is our cloud native platform that enables collaboration. We also have a website with extensive documentation to help our community adopt these tools. And also we support a forum where we interact with our users and help them optimize our pipelines for their purposes. And finally, some of the future work that we are working on is there's always room for improvement, and we want to continuously optimize and improve our GATK tools for bacteria. In addition to that, we also want to expand GATK to other microbes. Next, we will be applying GATK to fungal genomes, and then for viral genomes. And essentially, we want to package GATK in such a way that it can be applicable to any microbial species. And finally, I want to thank CZI for funding this project and for allowing us to build these robust tools for our microbial community. I see a question here about, do you think past use of GATK for bacterial data has been appropriate, or were enough assumptions being violated that we should cause be spectacle of results that predate your current projects? I think GATK out of the box works fine, but you would based on our analysis, we've seen that it does not have as good sensitivity as our current project. We've, in fact, not just sensitivity, we've improved on sensitivity and specificity and the precision.
All right. One question I had is, at least in my experience, sometimes when I'm working with an open source like research tool and then go to adapt it for a different use or a different community, it can be hard to kind of thread the needle between adapting it just enough that it does the thing that you want, while also like keeping users and stakeholders feeling grounded and people get stressed out by change. Kind of how have you approached that overall within your community, or is that something you've even encountered?
Let me restate that question. So how do we maintain, say, for example, benchmark the tools? Is that the question?
More or less, yeah. Well, and just helping people get their heads around, like Greg's question, how is this new work different from what it used to be? Like, what does that mean for my analyses? Do I need to adjust my workflow? All that kind of stuff. And I know you had some really great thoughts on kind of community outreach.
Yeah, and I think that has been key, not just for like what we're going to be doing next with GATK, but also for us to realize how GATK is being used, and the reason behind why we were able to do that is our interactions with our users. So we maintain our this forum, and we have dedicated efforts to support the forum, and every day we get about at least 10 questions about our tools, and we're constantly trying to solve all of these questions and help people optimize GATK for their purposes. And I think that and we also maintain extensive documentation about what changes we are making to the tools, how that would impact results. We share our benchmarking results. And I think all of that together keeps people informed about how the tools have changed, how these changes will impact their results.
Yeah, I mean, that's a huge amount of, more or less, yeah. I don't think I'm articulating particularly well. It's very early where I am. You'll have to forgive me. I'm still on cup 1 of coffee. But yeah, I mean, all of that is just a huge amount of work, and I'm so impressed that that the GATK development team is putting in all of that.
Okay, well, thanks everybody so much for coming. I'm going to talk about the QIIME microbiome bioinformatics platform. So this is a very widely used bioinformatics package for analyzing microbiomes. A lot of people are using this for human microbiome research, and so studying the literally trillions of microorganisms that live in and on the human body. Also very widely used for environmental microbiology, food microbiology, all kinds of things that we had never imagined when we first got started on this. And while my lab leads the QIIME development effort, it's really a huge global collaboration, and that's well illustrated by the author list on the primary QIIME paper, which we published a little over a year ago. There were 112 co-authors on this paper from 77 institutions in 10 different countries. And all of the folks on this author list contributed in some way to the design, the development, the documentation, or the support of QIIME. And so traditionally, QIIME has been thought of as a microbiome bioinformatics platform specifically focused on a particular data type, which I usually call marker gene data. And so that would be things like 16S ribosomal RNA surveys or ITS surveys that are essentially giving you a taxonomic or a phylogenetic profile of microbial communities. But just like how we think about GIS systems in terms of data layers, and so, for example, if we want to model the real world, we might have land usage and elevation and streets as different layers. We're starting to think about analyzing the microbiome using different data layers as well. And so in a microbiome survey, taxonomy, for example, might be one of our layers, but we might also want to know, say, like what functional potential is present in the community through a shotgun survey. And so, that might tell us about antibiotic resistance genes, for example. Or what functional activity is happening in this community, which we might get from transcriptomes or proteomes or metabolomes. And so really where we're moving now with QIIME is building this into a microbiome multi-omics bioinformatics platform. And that is some work that is now funded, and we're sort of in an early stage of development at this point. I have a post on the QIIME 2 forum linked here, and I have a link to these slides at the end if you want to learn more about that. But you might wonder how we're going to, so these are really different data types. Each of them has their own informatics needs. And you might wonder how we're going to achieve developing support for analysis of all these diverse data types. And the answer is, at least my team on its own is not going to do that. We're going to be tapping into that huge developer network that I mentioned in my previous slide. And so QIIME 2 is built based on a plugin architecture. So we have this core QIIME 2 framework, but all of the bioinformatics analysis functionality is implemented in plugins. The plugins, I sort of think of them as Python 3 method annotations that QIIME 2 interprets, but the underlying methods themselves don't need to be written in Python. In fact, some of our most popular plugins, the core functionality is written in R or is written in C, and then there's this Python layer that wraps it and makes it accessible to QIIME 2. And we already have a pretty big third-party developer community. The QIIME 2 library is our primary site for discovering and disseminating QIIME 2 plugins. And that site is really the focus of AIM 1 of our CZI grant. AIM 1 of this project is really focused on improving third-party developer support for QIIME 2. And so this is a work in progress right now. We have a relatively simple version of the QIIME 2 library that is now live, and it allows plugin developers to share their plugins and QIIME 2 users to discover new plugins. But we are in the process of implementing features in the QIIME 2 library that help plugin developers build, test, document, and release their QIIME 2 plugins. And so this is going to do a number of things. First, it will help it'll help developers keep their plugins up to date. We'll be doing things like automated testing, unit testing, and doc testing against development versions and release versions of QIIME 2. We do put a lot of effort into keeping the core API of the QIIME 2 framework stable. So it's not that often that things change. More frequently, it's things like versions of underlying dependencies that are changing. And so we will be able to alert plugin developers ahead of time, hey, your plugin is failing a test with the current development version of QIIME 2. And then that gives them some time to discover and adapt to the changes in the system. We'll also be doing things like building and publishing Conda packages for these plugins, and that'll remove that burden from the developers. The third-party developers. A lot of our third-party developers are not necessarily trained as software engineers. They might, for example, be statisticians who are working on building new methods. We find that building and publishing Conda packages is a stumbling point that we're often helping those developers with. And so we're working to just sort of simplify that and just help the method developers get their methods in the hands of our users more quickly. Now, there's a few benefits of QIIME 2 for plugin developers that I want to mention. And this will also sort of give you an idea of some of the cool things that we're doing with QIIME 2. So one of the things that attracts plugin developers to the system is that QIIME 2 has multiple interface options. And just like third parties can create plugins, third parties can also create interfaces. So QIIME 2 comes with a software development kit. And we expected to see we expected to see a lot of plugin development happening by third parties before interface development, which is exactly what happened. But we are actually now starting to see some third parties developing interfaces for QIIME 2, which is very cool. But the idea here is that a plugin developer writes the plugin once, and then QIIME 2 knows how to interpret that plugin and develop these different kinds of interfaces. So regardless of whether a user is like on the domain scientist end of the spectrum or on the data scientist end of the spectrum, there's an interface that is geared toward that type of user, and the plugin developer doesn't have to create it. QIIME 2 is also really focused on enabling reproducibility of bioinformatics methods and workflows. And it includes a pretty cool, pretty unique retrospective data provenance tracking system that facilitates reproducibility. And plugin developers don't even have to know that this exists. So they just, anything that their plugin is used for in QIIME 2 will automatically be recording data provenance behind the scenes. What this means is, for example, like a visualization like you see sort of on the left in the background there. That is a common visualization that people use. It's an interactive taxonomic bar chart. And a lot of bioinformatics steps happen before you get to that plot. Now, behind the scenes, QIIME 2 is recording all of those steps of the analysis. And so on this front panel that I have here, what you can see is that bottom box illustrates the current result that you're looking at. And all of the previous steps in an analysis are recorded here, including exactly what command was run, what parameters were provided, what inputs were provided, and what outputs were generated. There's information on the computational environment where this was run. So if you run some steps on a cluster and some steps on your laptop, all of that information will be recorded in those results. This is something that has it has become very popular with QIIME 2 users. It really helps us with providing tech support, and it really helps users document what they did and describe what they did. And again, plugin developers get all of this functionality for free. They don't have to think about supporting reproducibility in this way. We also have a very active online forum. We've got thousands and thousands of users on this forum. And so plugin developers can tap into this to provide and even receive support on the QIIME 2 forum. And so we support both users and developers on this forum. We get a lot of contributions from our community. Okay, the other thing that I wanted to go over in this talk was our QIIME 2 workshops. And so we regularly host workshops that are hands-on, that have networking opportunities and have small breakout opportunities for participants to interact and network with each other. We also teach third-party plugins in our workshops often, and we frequently try and get third-party developers involved in the events. And so that's another benefit that they get. But for our CZI project, we had planned to host a workshop that was going to be a new model. The pandemic, of course, disrupted that. We were not able to do our original plan, but we worked with the program officers and reworked that plan and held our first ever fully online workshop. And I just want to spend a few minutes just telling you about what we did here, because it was very exciting. So this was a 5-day fully online workshop, 6 hours a day. And it was, a lot of workshops are happening online now. A lot of conferences are happening online now. One thing that was really cool about what we did here was that this was focused around hands-on tutorials. And so that's what we do in person. We get a lot of good feedback on that. And so we wanted to figure out how to do a hands-on workshop online. And so we did this using an Amazon Web Services-based cluster that we deployed just for this event. And we ended up having 75 registered attendees from 6 continents and at least 20 different countries involved in the workshop. And so this was by far the most geographically diverse workshop that we ever hosted. We did charge a small fee, but we didn't actually need the funds really because CZI was funding this workshop. We just wanted the folks who were attending to be committed to actually showing up at the workshop. And so we ended up charging $50 for somebody to attend, but we also offered a purchasing power parity discount. And so we said when we started advertising this, that if you can't afford the workshop fee, reach out to us by email and we can work with you on that. We got a lot of requests for reduced cost. We approved all of them, no questions asked, even in cases where people said they couldn't pay anything for the workshop. And I think that really enabled this to be as diverse a group of attendees as we had. It was very exciting. We sold all of our seats for the workshop in under 5 minutes when we went live. So there's a lot of demand for this. In addition to having a very diverse group of participants, we had 21 instructors involved in this on 3 different continents, which was also enabled by the fact that this was an online workshop. And so the workshop itself consisted of some pre-recorded lecture content on bioinformatics theory, microbiome bioinformatics theory, some pre-recorded hands-on tutorials focused on putting that theory into practice, and then live Q&A sessions throughout the day with longer sessions at the end of each day. We also use Slack for communicating with attendees. All questions would come in on Slack, whether they were tech support or for the general Q&A. And so we triaged on Slack, and our instructors were able to work one-on-one with students, either by chat or by video, just depending on the type of question. And there were a lot of benefits to this online forum. So increased access for both the instructors and students was great. It sort of prompted us to create a ton of video content that we're now preparing to put on YouTube under Creative Commons, much lower carbon footprint than our in-person workshops. And so this was a unique opportunity created by CZI and by the pandemic, and we are definitely going to be doing more of these. We're working on writing up a workshop report right now. I'm just sharing some of the feedback we got here because I'm out of time. I won't go into this in detail, but we got really good feedback on this. We adapted our questionnaires from the Carpentries, and so I just want to thank them again for the great work and the great materials that they're providing. And I'll wrap up here by acknowledging our 21 instructors for this workshop. Again, these were folks from around the world. And I'll also acknowledge folks in my lab, the QIIME 1 and 2 communities, and the QIIME 2 funding sources. With that, I'd be happy to take any questions. It
looks like a pretty interesting project and a lot of overlap with some issues. I work on GATK with Banu. And I'm also doing my CZI project is related to HDS-GATK, which is a library layer underneath GATK. My question for you is, do you guys have any good solutions, or do you run into issues with consumers who want to use your software with multiple plugins that have different sets of underlying dependencies?
Yeah, it's, we haven't run into that a ton yet, but we're expecting it to become more and more common. And so one of the things, we don't have a great solution for this right now, but one of the things that we are going to do with the QIIME 2 library is we're sort of working toward an idea of like bundles of plugins. And so like if you want to deploy a set of plugins, you could handle that through different Conda environments, essentially. So like you could have some plugins that would, maybe have one set of dependencies, another set of plugins that might have a conflict somewhere in there. Right. And you could deploy that in different environments. And then the communication layer would be through the QIIME 2 artifacts, which are basically, they're basically like a container-based file that has the data in the usual format, like FASTA or NUIC or whatever, and then a bunch of QIIME 2 specific metadata. So it's not a great solution, but it's kind of what we're thinking right now.
Yeah, interesting. Thanks.
Yep.
Hi, this is Mary. I was actually really excited to hear about the workshop and the success that you guys had with that, and I'll be excited to read the workshop report. But I was wondering if there was any lessons learned as far as doing this workshop that you would do differently next time, or things that either, or conversely, things that worked particularly well for that.
Yeah, so in terms of lessons learned, probably the thing that what we were hoping to facilitate that we don't think worked out quite as well was networking among the participants. And so we have, in our in-person workshops, we call them water cooler chats. And so like if a topic comes up and we say like, there's it's too big of a topic to handle in a Q&A session, but we notice there's a lot of people interested in that. We try and direct them like during a coffee break to say, okay, over by, the back corner of the room, we're going to talk about ITS primers for, fungal surveys or whatever. And we tried to recreate that in Slack a little bit. And it didn't work out quite so well. So we're wondering if, we still had a lot of interaction among the attendees, but it was mostly on things like introductions, or like we had one channel where people were just sharing pictures of their pets. So like there's some fun stuff like that, but that's something that we're hoping to figure out. And I don't have a great solution yet. And actually it's one of the things that I'm kind of hoping to learn and see like how CZI handles that this week. We have another one of these online workshops coming up in January. And so we're looking for some new ideas to try out to facilitate that.
So with that, let's go ahead and thank Greg one more time. And then we will move on to our last presentation, last but not least, from the Salmon team.
Thanks, everybody, and thanks. Really excited to give a little overview of our the Salmon project. My name is Carl Kingsford. I'm a professor in the School of Computer Science in the Computational Biology Department at Carnegie Mellon, and also CEO of Ocean Genomics. And I'm really speaking on behalf of a much bigger team that has worked on the Salmon project, including Rob Patro and Guillaume Marseille, who I think are in the chat or in the Zoom meeting or soon will be. So what I'm going to talk about here is the work of a lot of people. So what is the Salmon project? It is an open source suite of tools to solve the gene expression quantification problem, which many of you are probably familiar with, but if not, the idea is that in order to measure how the level of expression of genes or transcripts, you obtain lots of sequencing reads in a technique called RNA-seq, and then those sequencing reads have to be translated into expression levels of genes or transcripts. And so what this really is a huge inference problem. We have tens of millions of reads. These are the observations, and we have, depending, 20 to 200,000 variables, which are the expression levels of the transcripts and genes that you want to infer, and that we have to fit. So we have what Salmon, the suite of tools, solves is this really big inference problem to try to estimate these expression levels. And estimating gene expression levels with RNA-seq is a hugely important biological assay. RNA-seq is the dominant method for measuring various aspects of the transcriptome, and one of the most important, which is quantification. And quantification is really the first step for a lot of tasks that enable things like biomarker discovery or gene function prediction or pathway analyses that identify, for example, which genes are expressed in people with a certain phenotype or not expressed in people with a different phenotype, and so on. So really, all that analysis starts with quantifying gene expression levels. And this is, it's one computational problem, but there's actually a huge amount of computational challenges and modeling challenges that have to be solved to solve it. The first is that reads can come from multiple locations because transcripts share a lot of sequences. So you have multi-mapped reads. So in addition to figuring out where the expression of all these transcripts, you have to figure out where the reads came from. There's sequencing and other experimental biases that have to be factored. There's a need for assessing and transmitting to the end user a measurement of uncertainty. And so people can know that certain estimates are very confident based on the data and certain estimates are less confident. And then, of course, there's the computational efficiency issue, which is how do we scale to huge numbers of experiments that people are now doing. And the Salmon project started when bulk RNA-seq sequencing was the norm. But the rise of single-cell sequencing, where instead of looking at a collection of cells, you look at individual cells and sequence them with various techniques, makes these computational modeling problems even harder to solve. So the Salmon project really looks for accurate, flexible, efficient solutions for that problem. And there are really two branches that are or sets of tools that are in the overall project. There's a set of tools for dealing with bulk RNA-seq and some single-cell RNA-seq data, and then another set of tools for dealing with tagged and single-cell RNA-seq data. And these sets of tools, the bulk RNA-seq and sort of the original approach is called Salmon, and the tagged single-cell RNA-seq protocol processing tools are called 11 and 11 Fry. And these components have been adopted in a lot of pipelines, downloaded 180,000 times via various distribution mechanisms. It's included as part of reproducible pipelines like NF-Core, which we heard the pitch earlier today, and ARMOR and others. And the single cell analysis have been used in some standard pipelines for the single cell atlas. So these are really important problems, and we're happy that people have adopted these things. And part of the reasons that we're excited to be a part of this CZI program, and also why some people have adopted these, is that we have developed the tools really from the start. You see some of the Git commits here from 6 years ago in the open. There's no hidden commits or anything. All the commits are open. We've gotten user feedback and input from various users from the beginning of the software. And we tried to support a vibrant open source community with a lot of interactions across GitHub, BioStars, Google Groups, Gitter, and so on. And also one of the things I'll talk about later is how we can improve this community. So to give you a little bit more background on the goals of the Salmon project in terms of high-level goals, it really focuses on these 5 things: accuracy, accurate solution of that inference problem, efficient solution, flexible usability, reproducibility, and interoperability. And in terms of accuracy, over the years, we've improved, we and the team has improved this software in accuracy in a number of ways. First of all, introducing fragment level bias modeling, and then improving the objective function that the software solves so that you improve its accuracy without slowing down the performance. You can use a more complicated, more accurate likelihood model. And then also looking at how the ways people provide input to the tools affect the accuracy, and developing new ways of mapping reads to locations in the genome, such as selective alignment. Selective alignment is a lightweight alignment strategy that lets you get the accuracy from doing like a full alignment of your reads to the transcript tome, but with the speed of a lightweight alignment. efficiency. A number of data structures have been developed and incorporated into the tool to improve its speed. Here's one example of them to store indices that the software has to make more efficiently. These kinds of advancements allow the tool to process huge data sets with significantly faster than other approaches. Twice as fast with a third as much memory for a 10,000 cell single cell data set, for example. The software really aims to be kind of able to be dropped in many different settings. So it accepts either aligned BAM files, so you can do your alignment, map your reads to the transcriptome of interest, provide that as input, or you can give it just the raw FASTQ files directly off the sequencer, and Salmon will do its selective alignment and produce these estimates. Okay. Salmon's integrated in a number of workflows that enable reproducibility. Here's an example of workflow or a system that enables reproducibility by tracking the where your quantified transcripts and results came from. And Salmon is the first and currently only quantification tool in this system. And there's a huge amount of downstream packages that quantification with Salmon kind of feeds into, a number of which are supported by the CZI or others. And here I won't go through all of these, but there's packages for differential gene expression, differential transcript usage, single cell analyses, single cell clustering, and so on. So we really try to interoperate with all of these as best as possible. So just in the last couple of slides, I wanted to talk a little bit in more detail about where we see things going, particularly in terms of this CZI project. The overriding goals of the project are to expand the types of inputs from a technical level that Salmon can accept, extend the algorithms to support more data types, improve the documentation and create training materials so that more people can use the software, and to continue to build and create an even more vibrant community around the software. And just in a little more detail, what do I mean by expand the types of data or input that they can accept? Here is on the left 5 bullet points that are specifically talking about things that are important for making the software easy to use, make it easy to reanalyze large collections of data, make it easy to reuse existing collections of publicly available data, but are now, there's a sort of a speed bump or a pain point where users can't do that easily because the software doesn't support some particular type of input. And so, for example, we are going to implement reading from genome-aligned BAM files rather than having your reads aligned to a transcriptome. You can align your reads to the genome. This allows you to reuse this alignments that you may be using for other things like transcript assembly or variant calling, accepting input from unaligned BAM files, reading files from the NIH SRA sequence read archive directly, automatically performing adapter and quality trimming via local alignment so that users don't have to do an additional step, which many users often omit or don't do correctly, and thus reducing the quality of the results, and support more single cell protocols, particularly highly multiplexed ones and protocols with variable length barcodes, UMI and UMIs. And these are, some of these are interesting algorithmic problems, and some of them are interesting engineering problems. For example, you might think accepting genome-aligned BAM files is just writing a new file type reader, but there's some technical challenges, which is that Salmon has to process the paired ends of the reads together, but those may be very far apart in the BAM file. And also Salmon, because it's it's a randomized inference algorithm, assumes that the reads are in random order, which would not be the case in a sorted BAM file. Okay, so there are some challenges in terms of how to implement them, and that's what makes this interesting. But the end goal is to enable the ability to reprocess and process larger and larger collections of experiments. And then the last major goal is really creating additional training materials and improve the community engagement. And there are particularly 11, but Salmon 11, 11 Fry all have some tutorials that have already been created. On the right of the slide here is the beginning of one that looks at doing spatial transcriptomics with 11. But there's a huge number of use cases that people are interested in applying these tools to where there's not good documentation. And so we're going to create additional tutorials, common workflows, and training materials in things like video and other mediums. Okay. And that's it for me. Thank you, of course, to the Salmon team, who I'm really speaking for, and to the users and contributors to the Salmon project. Thanks to CZI for providing funding. We're very excited to be part of that team. And thanks everybody for your attention. And at the bottom here are links to the GitHub repos and our Twitter. And please reach out if you have suggestions or ways to interact with us. So, thank you.
My question, and please forgive me if I missed this bit, but can you comment a little bit on how Salmon integrates with the rest of the ecosystem, like computational ecosystem for processing single-cell RNA-seq data?
Yeah, actually, I don't know if Rob can jump in on that. He's in charge of the single-cell aspect. Are you there, Rob?
Yeah, I am here.
I put you on the spot, but maybe you could answer that.
Yeah, sure. No, absolutely. So, Salmon, so specifically Alvin, and recently Alvin Fry, which is sort of a new improved version of Alvin that we've been developing, interface with the single cell ecosystem in a number of ways. But the primary one is there's two different packages. So the input to R is currently right now through TX import and TXI meta. So these are packages developed by and in conjunction with Mike Love's lab and a number of others that make importing 11 and 11 Fry output into R easy, and then provides sort of standard R data structures like summarized experiments and things of that nature for downstream use. And then in single cell in particular, ScanPy has become pretty popular for folks who want to use Python. And for that particular pathway, we actually maintain a Python package developed by a former graduate student of mine, is available on Bioconda and pip, called vPolo, which is for importing the output of 11 and 11 Fry into Python data structures for downstream use with ScanPy. So we actually provide a couple of different output formats. Some of them are focused on compression and keeping the output files small, and then others are focused on easy parsability. So things like a standard sparse matrix format.
Mm-hmm. Fantastic, thank you. Yeah, the vPolo bit was the part that I missed.
Yeah, sure.
Awesome, thank you so much.